Okay, good afternoon. I think we will begin. We have a hard-earned reputation for doing our events on time, and out of respect for you, we will maintain it. Welcome, everyone. My name is Joseph Kearney, and it is a great privilege for me as Dean of Marquette University Law School to welcome you to our annual Nies lecture in intellectual property. We really are delighted that you are here. Let me begin by recalling the person in whose memory this lecture stands. The law school created this lecture in 1998 to honor the late Helen Wilson Nies, who had served as a judge of the United States Court of Appeals for the Federal Circuit, including a term as its chief judge. We did this at the suggestion of one of our faculty at the time. Judge Nies is widely remembered within the intellectual property bar as a very generous individual and as a hardworking, serious, and modest judge. For the past 2 decades, it has been a great privilege for us to make this lecture a constant of our intellectual property program, even as, under the leadership of Professors Bruce Boyden and Kaylee Murray, we have been able to expand that program in recent years. In addition to actually having known Judge Nies, as perhaps we'll hear, today's lecturer is a leading scholar of intellectual property law. Rebecca Eisenberg is the Robert and Barbara Luciano Professor of Law at the University of Michigan, where she has served on the faculty since 1984. She teaches courses about patent law, trademark law, international intellectual property law, and FDA law. Professor Eisenberg has lectured and written extensively about the role of intellectual property in biopharmaceutical research, publishing in leading law reviews and scientific journals. She also has played an active role in public policy debates concerning intellectual property, advising the National Institutes of Health and the National Academies of Science. She is a graduate of Stanford University and Bolt Hall School of Law at the University of California, Berkeley. Today, she is with us. Won't you please join me in welcoming, for this year's Nies lecture, Professor Rebecca Eisenberg.
Thank you very much. It's a great personal honor for me to be here to deliver the Helen Wilson Nies lecture in intellectual property because she is somebody who is meaningful to me. I want to say a few words about Judge Nies, who was a distinguished alumna of the University of Michigan Law School, class of 1948, before there were a lot of women in ours or any other class, and we are very proud of her. I had the pleasure of getting to know her some years ago when she returned to her alma mater and spoke to my patent law class. She was, of course, an outstanding student, graduated Order of the Coif, and got a prestigious job in the Justice Department straight out of law school. But after taking a few years off to raise her kids, she discovered it wasn't so easy for women to re-enter the job market. She had some stories to tell, but she obviously managed quite well. She had a distinguished career in trademark law before she was appointed to the federal bench in 1980, and with the creation of the Court of Appeals for the Federal Circuit in 1983, she became one of its first judges and eventually served as chief judge. We tend to think of the Federal Circuit as a specialized patent court, and Judge Nies had little background in patent law before she was appointed to the bench, although her husband, also a Michigan law grad, was a patent lawyer. But of course, patent law is law, and Judge Nies was an excellent lawyer, so she rolled up her sleeves and she figured it out. I admire that fearlessness, especially at a relatively late career stage. None of us really knows where our careers will take us. And if we want to take advantage of new opportunities, or even if we just want to give our clients the advice that they need as the world changes, we may need to roll up our sleeves and learn something new. Among Judge Nies's many important contributions to the Federal Circuit were some early opinions on legal process and procedural issues that the new court needed to address in order to serve its mission of consolidating appellate jurisdiction over patent law and standardizing its interpretation. And her opinions on burden of proof, standard of review, and the authority of prior opinions and of other courts continue to be cited to this day, while many many substantive decisions of her Federal Circuit brethren from the same era have been overturned by subsequent decisions of the Supreme Court. The enduring significance to the patent system of these process issues reminds us that patent law is not an island apart from the rest of the legal system. It works alongside other bodies of law, and it operates through rules of administrative and judicial practice that are not unique to patent law. And I've thought a lot about Judge Nies over the years, as I found it increasingly necessary to look outside patent law in order to understand the environment in which patent law does its work. The primary focus of my scholarship has been biomedical innovation. Patent law often gets much of the credit for motivating investments in biomedical innovation, particularly from the pharmaceutical industry, which has worked hard to strengthen patent laws throughout the world. But patent law does not do this work alone. And this is a lesson that I keep learning over and over again. Today, I want to examine the intersection of patent law, FDA regulation, and Medicare coverage in a particularly promising field of biomedical engineering, genetic diagnostic testing. This lecture has a lot of moving parts, so I want to start off with a roadmap. You may also want to follow along on the outline if you like visual aids. I have no slides for you today. First, I'm going to talk briefly about genetic testing today, its current clinical uses and directions for further research, with a focus on cancer, which is the field in which it's had the greatest impact so far. I will then turn to patent law and tell you about recent cases that call into question the patentability of some of the most important advances in genetic testing. This is how I came to the topic as a patent scholar, reading these cases and worrying, what does this mean for important innovation that remains to be done in the area of genetic diagnostics. Then I will step outside patent law to take a broader view of the legal environment for new developments in genetic testing, with a focus on two federal regulatory agencies: the Food and Drug Administration, or FDA, which regulates, of course, new drugs and medical devices under statutory standards for safety and effectiveness, and the Centers for Medicare and Medicaid Services, or CMS, which sets reimbursement policies for Medicare under statutory standards that limit Medicare coverage to technologies that are reasonable and necessary. I will then turn to some recent developments that took me by surprise and try to explain why developers of tests that use next-generation sequencing technology to sequence large amounts of DNA from cancer tumors have begun seeking FDA approval for tests that they don't need FDA approval to sell, and in fact that they were already offering without FDA approval fully lawfully. Spoiler alert, at this point we will need to digress to understand the rules and practices that govern health insurance coverage. So I think this episode sheds an interesting light on the role of different sources of legal regulation in supporting innovation, even in the biopharmaceutical heartland, which we think of as the paradigm case for the importance of patents in motivating innovation. The science is fascinating. Advances in genetics and molecular biology have transformed scientific understanding of the basis of many diseases, identifying new molecular targets for therapy and rearranging diagnostic categories that shift in ways that shift our understanding of disease. Nowhere is this more striking than in cancer. Traditional cancer diagnosis focuses on the tissue of origin of a tumor, with different treatment options coming into view depending on whether the patient has breast cancer or colon cancer or lung cancer, et cetera. This is the way we all talk about cancer. We start with the tissue of origin to specify what type of cancer a patient has. But increasingly, it looks like what really matters is not so much the tissue of origin as the genetic mutation that is driving the tumor. What went wrong in a cell that caused a tumor to develop. Advances in understanding cancer at the molecular level have revealed a few hundred genes in which mutations arise that are associated with cancer. Some of these mutations are common and well understood, while others are rare, and their role in driving cancer remains unclear, even though there is a recognized association. Drug companies have been hard at work at developing a new generation of targeted therapies that are designed to work against tumors with some of these mutations. Sometimes these therapeutic products are developed and submitted to FDA for approval along with a companion diagnostic to test whether a patient's tumor has the targeted mutation. The FDA-approved indications for these targeted therapeutics may specify a tissue of origin as well as a particular genetic mutation that is targeted by the therapy. So, for example, one of the earliest of these drugs was Herceptin, which is a cancer therapy. The original FDA-approved label for Herceptin specified that Herceptin is a HER2 new receptor antagonist indicated for the treatment of HER2 overexpressing breast cancer. Later, they discovered, the same gene seems to sometimes be implicated, the same mutation sometimes seems to be implicated in other kinds of cancer. And so they expanded the label to include the treatment of HER2 overexpressing metastatic gastric or gastroesophageal junction adenocarcinoma. So they added a new tissue type along with the original one. Last summer, for the first time, FDA took a much bigger step, not for Herceptin, but for an immunotherapy drug called pembrolizumab, perhaps I'm mispronouncing that, expanding the approved indications to include all tumors with the specified genetic profile, regardless of the tissue of origin. So we may be moving towards a future in which the essential piece of information for meaningful diagnosis and for selecting appropriate treatments is not the tissue of origin, but genetic testing. Genetic testing is going to become an important first step in sorting out patients and figuring out how best to care for them. Much work remains to be done to understand which mutations are meaningful. Meanwhile, the cost of genetic testing has come down dramatically. A big factor has been reductions in the cost of DNA sequencing. Many steps have become automated, and using so-called next-generation sequencing techniques, it is now possible to sequence a large amount of DNA, even perhaps a whole genome, in a single test for a few $1,000s, which, when you consider the cost of cancer care, is pretty trivial. And this changes how it makes sense to proceed with genetic testing for cancer patients. When Herceptin was first approved for treatment of HER2 new overexpressing breast cancer, it seemed sensible enough to test tumors from breast cancer patients for just the particular aberration that was targeted by that drug to figure out which patients should be treated with Herceptin. But today we have a much larger set of targeted therapies against different mutations, and we know a lot more about the genetics of cancer, and it's a lot cheaper to get a lot more information out of a single test. So for a modest incremental cost, it's now possible to fully sequence the 350 to 400 genes with a known association with cancer in a DNA sample from a patient's tumor, rather than just to test for a particular targeted mutation. And this can spare the patient the need to come back for multiple tests when one drug doesn't work. A number of different laboratories have these tests available, including some in academic medical centers and some in commercial firms. So this sounds like a great R&D success story, and a surprising one from my perspective as a patent scholar. Why did I find this surprising? Well, because recent judicial decisions about patentable subject matter from the US Supreme Court have cast serious doubt on the patentability of many of the most important advances in DNA diagnostic testing. Two US Supreme Court decisions in particular cast a pall over these patents. The first was a 2012 decision in a case called Mayo versus Prometheus that some of us spent some time together considering. The patent in that case is an example of a patent on a diagnostic algorithm that involves observing a particular biomarker, which could be a DNA sequence or it could be the level of a certain protein in a patient's blood, and then drawing an inference, a diagnostic inference about the patient's need for treatment. More specifically, in that case, the patent claimed a method of optimizing treatment with thiopurine drugs by measuring levels of certain drug metabolites in a patient's blood and determining on that basis whether the dosage of the drug needs to be adjusted. Metabolites are the products that the patient's body produces as it breaks the drug down after ingesting it. The patent recited both a lower level of a particular drug metabolite that would indicate a need to increase the dosage to make sure that it was therapeutically effective, and an upper level that would indicate a need to dial it down a bit in order to avoid toxic side effects. That was basically what the claim was about. And the Supreme Court held that this patent claim was invalid because the metabolite levels that would indicate a need to adjust the dosage of the drug are unpatentable laws of nature. And that once you exclude those laws of nature, there wasn't enough else left in the claim to give rise to a patentable invention. I think this is, a fair reading of the court's analysis would seem to count as natural laws all predictions of the effects of treatment in patients. It might sound like I'm exaggerating a bit. For a while, perhaps there was some hope that subsequent decisions would interpret this more narrowly, but that is not what has happened. It seems that the broad reading is the one that has prevailed. I don't want to dwell on this case, but in the interest of full disclosure, I should probably tell you that I think the court is fundamentally confused here, failing to recognize medical diagnosis as a human technology rather than a mere observation of the natural world. Nature doesn't tell you when you need more or less thiopurine drugs. Nature doesn't administer thiopurine drugs, and it doesn't say anything about how to trade off the benefits of thiopurine treatment against its side effects. These are human judgments that are part of the human technology of medicine. Anyhow, this was a major blow to patenting strategies for diagnostic tests. A year later, the Supreme Court dealt another major blow with its decision in Association for Molecular Pathology versus Myriad Genetics. The patent at issue in that case claimed DNA molecules with sequences corresponding to the BRCA1 and BRCA2 genes associated with susceptibility to developing breast cancer. The Supreme Court held that these genes, as they exist in patients' cells, are unpatentable because they are natural products. And a fair reading of the court's analysis seems to count as natural products all the DNA biomarkers used in genetic testing. Again, maybe you could read them more narrowly, but that has not happened in subsequent lower court decisions. Putting these two decisions together, there is now considerable doubt about the patentability of both the biomarkers and the diagnostic algorithms or predictions that are involved in genetic diagnostic testing. That's basically what this science is about. It's about identifying markers and correlating them with predictions. So what does this mean for incentives for the investment in the development of genetic diagnostic tests? Well, if you look to the conventional wisdom for the biopharmaceutical industry, you might worry that patent incentives are essential and that investment should therefore dry up if the patentability of these inventions is called into question. I should also note, however, that these Supreme Court decisions were, lamented by patent folks were much celebrated by other people in the field of genetics research, especially in academic medical centers where they felt like these decisions were freeing them to perform genetic diagnostic testing that they had previously been unable to do because of fear of patent infringement liability. Particularly if they wanted to offer these comprehensive tests, these like next generation sequencing, let's look at 350 to 400 genes, without having to worry about being liable to multiple patent holders. So I had to consider the possibility that, maybe genetic testing is different from the paradigm case of new drug development that has long been the sort of go-to example of the need for patents to preserve incentives for new product development. Maybe there are differences between diagnostics and therapeutics that make patents less necessary for development incentives in the diagnostic space. One difference has to do with the regulatory burden imposed on these two types of products. The costs and risks of FDA regulation loom large in standard accounts of why the pharmaceutical industry needs patents on new drugs. Before a drug company can sell a new drug in commerce, it needs FDA approval for safety and effectiveness based on data from randomized controlled clinical trials that meet stringent scientific criteria. And not only is it very costly to conduct these trials, but there is a considerable risk of failure at every stage. Even after approval, many of these drugs get withdrawn from the market after new risks come to light. So drug developers argue, plausibly, that they need to cover the costs of all of these failures with the prices that they can charge for the occasional success story, and they need patents in order to be able to charge those elevated prices in order to make that business model work. But maybe genetic tests are different. FDA has statutory authority to regulate genetic tests for safety and efficacy as medical devices, but FDA regulates medical devices with a lighter touch and under different statutory provisions that encourage it to do this. Medical devices are a diverse category that ranges from things like tongue depressors and bandages to cardiac pacemakers. Some of these pose minimal risks and don't really need much regulation. Others need a lot of regulation. The statute directs FDA to take a stratified, risk-based approach to device regulation that sorts devices into three categories, Class 1, Class 2, and Class 3, according to the kinds of controls that are necessary to ensure their safety and effectiveness. And only the riskiest Class 3 devices require pre-market approval from FDA before they may be sold, while intermediate risk Class 2 devices may be sold after a less onerous pre-market clearance process, sometimes called the 510K process or 510K clearance after the relevant statutory provision. Even for the Class 3 devices that require pre-market approval, the burden and cost are considerably less for devices than they are for pre-market approval of drugs. FDA, for example, relies less heavily on pre-market testing and more on post-approval monitoring and data collection to ensure safety and effectiveness for drugs. So where do genetic tests fit into this scheme? Well, sometimes FDA regulates particular genetic tests as companion diagnostics for a new drug, as I was mentioning earlier, and will approve both of them together. Submission of data on a diagnostic, a genetic diagnostic for the targeted mutation, along with the targeted therapy itself, may allow the new drug to get approval more readily after clinical trials that are smaller and shorter in duration, and that focus on a subset of patients that have been selected because they are likely to respond to this drug, which makes the results look really good. So drug companies may be highly motivated to do the necessary testing to validate these particular tests for these particular mutations, because validating the tests will allow them to get approval to sell the new, presumably patented, drug that targets that mutation. So far, FDA has classified these companion diagnostics as class 3 medical devices requiring pre-market approval. But FDA guidance documents have indicated that, some companion diagnostics might be class 2 devices. In future cases, we might be willing to down-classify them if they determine that the class 2 controls, pre-market clearance and notification, are sufficient to provide a reasonable assurance of safety and efficacy. And this is consistent with FDA's flexible approach to device regulation. They often initially classify a new type of device in class 3 because they don't know that much about it. They haven't had that much experience with it. And then they'll require initially pre-market approval. Then they'll reclassify it later as their initial concerns about safety and effectiveness prove to be manageable with less burdensome regulatory controls. So it may be that even for these companion diagnostic tests, the level of regulation is lower. There's another important limitation on FDA regulation of diagnostic tests that so far has provided a huge break for these new comprehensive DNA tests, which is that as a matter of administrative discretion, FDA has chosen not to require either approval or clearance for what it calls laboratory-developed tests. A laboratory-developed test is a diagnostic test that is designed, manufactured, and used all within the same laboratory, rather than sold or licensed for use in other facilities. This is an old policy that was originally designed maybe for simpler tests, but it has allowed many genetic testing laboratories to provide testing services without having to go through FDA at all. A few years ago, FDA appeared to be on the verge of reconsidering this discretionary policy. They issued draft guidance in 2014 proposing to use expert advisory committees to sort out these laboratory-developed tests into different categories and to enforce their existing authorities to regulate the highest risk products more aggressively. But in the final days of the Obama administration, FDA decided to put these plans on hold. And the Trump administration has not shown any inclination to pick up where they left off. So it seems, for now at least, most laboratories that perform genetic testing services in the same laboratories that developed and manufactured the tests don't need approval or clearance for their tests from FDA. When FDA approval has been sought for a particular genetic test, it has generally been a specific companion diagnostic product developed in tandem with a drug that is specifically targeted against a genetic mutation and that might be performed by multiple different labs and therefore won't qualify as a laboratory-developed test. Meanwhile, these more comprehensive genetic tests that use next-generation sequencing to examine hundreds of genes that may have mutations driving a patient's cancer, they have become available without the need for FDA approval. And these tests have proliferated in recent years, some from academic medical centers and some from commercial labs. So again, this looks like a happy story, right? Of productive R&D, bringing new technology to market despite considerable uncertainty about the availability of patents. And we can explain this fairly readily. A couple of stories come to mind that seem to have a fair amount of explanatory power. First, maybe we can explain the willingness to invest in laboratory-developed tests by FDA's exercise of discretion not to regulate these products, at least so far. And second, as for the companion diagnostic products, we can explain the willingness of firms to invest in these products by the fact that having validated diagnostics helps these firms develop lucrative new patent-protected drugs targeted against specific mutations and get those patent-protected products approved. In both of these stories, firms are motivated to avoid the costs of FDA regulation and are more inclined to invest in the face of lower regulatory costs and risks. So these stories are also consistent with broader narratives about costly regulation as a drag on innovation. But those stories don't explain an interesting development over the past year or so that suggests maybe there's trouble in paradise here. Maybe this isn't as happy a story as I've been presenting. Laboratory laboratories that offer genetic testing of tumor DNA have begun seeking FDA approval of their products, even when they qualify as laboratory-developed tests for this FDA policy of administrative discretion to just let them go. These laboratories are free to offer these tests without FDA's blessing, and in fact, they were already lawfully operating their tests without that blessing, nonetheless voluntarily decided to submit these products to FDA. And this past November, FDA approved 2 very similar next-generation sequencing tests that detect mutations in hundreds of genes in tumor DNA samples. One application was from Memorial Sloan Kettering Cancer Center, and they sought FDA clearance of their test as a Class 2 device. The other was an application from a private firm, Foundation Medicine, for pre-market approval of its Foundation 1 test as a Class 3 device. The choices of these different regulatory pathways have interesting consequences that I'm going to come back to in just a minute. But first I want to pause and consider, why would these laboratories take upon themselves the costs and risks of submitting their products to FDA regulation when FDA does not require it? And that brings us to health insurance. The short answer is that health insurers were refusing to pay for their tests. This itself is a bit of a puzzle, since the cost of testing is trivial compared to the overall cost of cancer care. So insurers that readily pay in excess of $100,000 a year, and more really for some of these new targeted therapies, for expensive cancer care, would decline to pay a few $1,000 up front for testing that might reveal in a single test what is driving the cancer and whether the patient is a candidate for any of, 15 or so previously approved targeted cancer therapies that are already clinically available. What the insurers say is that they are willing to cover less comprehensive tests that focus on clinically validated mutations for cancer that have been shown to predict treatment response in patients with cancer of that type, but not the more informative tests that sequence more DNA and are likely to reveal not only the particular druggable mutations that have associated approved targeted therapies available, but also other mutations of unknown significance in hundreds of genes. Even though the difference in cost is trivial, insurers see an important principle at stake, which is that they don't pay for research, or they don't want to pay for research. Actually, they pay for quite a bit of research, but they resist. And they have a point. There is immediate clinical value in genetic testing to identify candidates for targeted therapies, but there's also considerable research value in detecting other mutations in genes that are known to play a role in cancer. Tracking these mutations in registries of cancer patients, along with their health records, would provide valuable data for researchers seeking a better understanding of cancer, perhaps enabling future improvements in cancer treatment. It would also provide a check on whether when cancer treatment is working and when it's not. It would allow you to correlate patient responses to tumor DNA variants. In other words, genetic testing has great research value as data collection. And insurers have a tradition of not paying for research, at least as a formal matter. Now, in fact, insurers pay for lots of research. FDA does not regulate the practice of medicine. It only regulates new drugs and devices. And so many innovations in healthcare have their origins in clinical care that has not been rigorously tested before it entered into clinical use. If it's a really expensive procedure, the insurance companies might resist. So, for example, autologous bone marrow transplantation for cancer patients, or lung volume reduction surgery for COPD. But much experimental care is going to fly below the radar of the insurance companies, and so as a practical matter, much biomedical innovation depends on insurers providing coverage of innovative new care, and they have long paid for it. And insurance coverage is especially important to facilitate innovation in areas that are not regulated by FDA, because FDA regulation means that somebody has got to do the pre-market testing that will get FDA approval before anybody's in a position to ask their insurance company to pay for it. But if FDA isn't regulating in an area, like a new medical procedure, the data may never come into being. And therein lies the catch-22 for unregulated cancer genetic testing, which is that insurers won't pay for testing unless the results have validated clinical significance. Drug companies will pay for pre-market validation of the relative handful of mutations that will allow them to get targeted therapies approved by FDA, but beyond these druggable mutations, they have less interest in the larger project of understanding the clinical significance of the larger universe of variants in genes that are known to play some role in cancer. And because many of these variants are quite rare, it's not really feasible to study them in the sort of pre-market clinical trials that drug companies are in a position to conduct, that they typically undertake in pursuit of FDA approval. You need to track much larger populations in order to get enough data to assess clinical significance for rare variants, and that's better done in observational studies in the course of clinical care. But clinical care won't happen without insurance coverage. So validation requires use in clinical care, use in clinical care requires insurance coverage, and insurance coverage requires validation. That's the catch-22. And this dilemma, I think, highlights an important function of FDA regulation that helps explain why innovators would seek FDA approval for new technologies that they are free to market without that approval. FDA performs a technology assessment function that public and private insurers rely on in deciding what to pay for. For public insurance, such as FDA, there's some federal law that determines what they are authorized to pay for. Federal law authorizes payment for reasonable and necessary care for Medicare patients. Regulations from CMS interpret this language to exclude experimental care. The private insurance policies often include similar language, and private insurers will often copy whatever Medicare is doing in deciding what they will cover. Everybody is trying to economize on technology assessment because technology assessment is difficult and costly. Now, reasonable and necessary care under the Medicare statute is not necessarily the same thing as safe and effective under the Food, Drug, and Cosmetic Act. Nonetheless, for the most part, health insurers provide coverage of FDA-approved technologies, although they may require prior authorization in some cases when cheaper alternatives are available. Sometimes insurers are required to cover these products by law under federal or state law coverage mandates that the CMS and the Trump administration is gradually issuing waivers to relieve them from. And sometimes they are simply avoiding the burden of conducting their own technology assessment by relying on FDA's determinations, which are close enough. This is a significant benefit of FDA approval that may go far to explain why Foundation Medicine and Sloan Kettering would voluntarily submit to FDA regulation, even though they are not required to do so. Presumably, they hoped that FDA approval would serve as a good enough proxy for clinical utility to persuade insurers to pay for testing. This is particularly clear in the case of Foundation Medicine, which was using a coordinated parallel review process in an effort to get simultaneous FDA approval and CMS coverage. So how's that working out for them, you may be wondering. Well, it's working out better for Foundation Medicine than for Sloan Kettering. Remember I mentioned earlier that these two institutions pursued different regulatory pathways for similar tests at FDA, and I promised to come back to that. So here I am. Foundation Medicine chose the more arduous pathway. They sought FDA approval of their test as a Class 3 medical device, calling it a companion diagnostic that was intended to be used to identify patients who may benefit from treatment with a list of targeted therapies that FDA had previously approved in accordance with the FDA-approved labeling for those products, as well as for tumor mutation profiling by qualified healthcare professionals in accordance with professional guidelines. Sloan Kettering did not call their very similar test a companion diagnostic. Instead, they called it a tumor profiling test that is intended for use by qualified healthcare professionals in accordance with professional guidelines and that is not conclusive or prescriptive for labeled use of any specific therapeutic product. They asked FDA to classify the device into Class 2, allowing it to be marketed upon compliance with less burdensome submission requirements under the pre-market notification process, that 510K process that I was talking about earlier. To approve this classification, FDA had to make a determination that the lesser regulatory controls applicable to Class 2 devices were sufficient to ensure that the device was safe and effective. And FDA ruled in favor of each of these applicants. They gave each applicant what it sought. But the difference between Foundation's pre-market approval of its test and the pre-market clearance for the Sloan Kettering tumor profiling test seems to make a big difference to CMS. On the same day that FDA approved the Foundation 1 test as a companion diagnostic, CMS issued a proposed national coverage determination for next-generation sequencing for Medicare beneficiaries with advanced cancer. Although they were responding to a request for determination by Foundation Medicine, they decided that they would more broadly address coverage for all of these next-generation sequencing tests in a single national coverage determination. And under Part A of that proposed decision, CMS would authorize full Medicare coverage of NGS testing for Medicare beneficiaries with advanced cancer for if the test is an FDA-approved companion in vitro diagnostic and used in a cancer with an FDA-approved companion diagnostic indication. That right there is enough to do the job for Foundation Medicine because their test is an FDA-approved companion diagnostic. But it's not going to be enough to do the job for Sloan Kettering because their test is a tumor profiling test, not a companion diagnostic, and because they sought approval as a Class 2 device that was technically cleared through a pre-market notification process rather than FDA approved. So if you read it quickly, you might not realize. I had to read it several times before I recognized what exactly was going on here. For products that are not companion diagnostics and that are merely FDA cleared and not FDA approved, CMS proposes more limited coverage under Part B of its proposed decision. That more limited coverage authorizes what CMS calls coverage with evidence development, or CED, which is an interesting innovation that provides Medicare funding for some experimental technologies for patients who are enrolled in clinical trials or data registries. The idea is that we're not going to provide full coverage yet because we're not really sure if this works, but we're going to provide coverage for people who are enrolled in trials or data registries so we can come back later and figure it out and decide whether to cover it or not. It's a really good idea, especially for technologies that are not FDA regulated and therefore arrive and become available for use and potential coverage before you really know whether they work or not. See, CED has been controversial among bioethicists because it could have the effect of coercing patients into participating in clinical trials. But I think it's a really interesting device, and it's a departure from the traditional refusal of insurers to pay for research that has real promise as a mechanism for collecting data to permit insurers to do more of their own technology assessment on the effects of healthcare technologies in clinical use. So far, it has mostly been used for procedures that don't go through FDA approval at all, like the examples I was mentioning of autologous bone marrow transplants for cancer patients. Sometimes the result has been to generate data that prevented costly and toxic new technologies from becoming the standard of care, because it turned out that these things didn't work so well in some of the patients in whom they were used. CED can also provide a solution to the catch-22 problem I mentioned earlier, in which data collection on a meaningful scale can't happen without coverage, because you need widespread utilization that can only be achieved through insurance coverage. So I'm a fan of CED, and I would like to see its use expanded as a general matter. On the other hand, if I were Sloan Kettering, I would surely feel bent out of shape by this decision. I would feel that it was unfair for CMS to be providing full coverage of the Foundation 1 test and only CED for the very similar and arguably superior, in fact, Sloan Kettering test. The distinction seems particularly unjustified, given that the same standards for safety and effectiveness apply to FDA-cleared Class 2 devices as apply to FDA-approved Class 3 devices. The difference in classification just turns on what controls FDA thinks are necessary in order to ensure safety and effectiveness for different types of devices, not whether the devices are or are not yet shown to be safe and effective. Moreover, the result of the different treatment for these two similar tests on the basis of which FDA regulatory pathway that the innovators chose could have a perverse consequence for future test developers. That is, if they hope to get insurance coverage, they may have to choose the more onerous FDA approval pathway for Class 3 devices, thereby undermining the efforts to streamline the process of regulatory approval. Many people submitted comments to on the CMS proposed decision, including me. So maybe they will revise it in the final national coverage determination. We'll see. I think this is a fascinating case study of the interacting regulatory regimes that set the stage for biomedical innovation. Patent law is often an important part of the picture, but not always. With or without patents, you won't make any money unless people buy your product, and people won't buy new technologies unless they believe they will be useful to them. In the healthcare context, this means that you need to persuade caregivers to prescribe the product, and you need to persuade insurers to pay for it. Patent law has nothing to say about the demand side of new technology development. That requires some technology assessment. FDA approval performs a technology assessment function that makes doctors more willing to adopt a new technology and makes insurers more willing to pay for it, even when FDA otherwise does nothing to stop the technology from reaching the market. And we can see in the proposed CMS coverage determination how much deference they are ready to give to fine distinctions in FDA technology assessment. Finally, if insurers will only pay for FDA-approved technologies, the need for FDA approval creates a costly entry barrier that slows down the entry of competitors. This starts to look a little bit more like a patent, right? It's it doesn't last as long as a patent, maybe, but for the moment, only one of these comprehensive companion diagnostics meets Medicare's requirements for full coverage. Presumably, others will eventually enter, but that's going to take time. Meanwhile, Foundation Medicine is enjoying the benefits of de facto market exclusivity, even without patents. So you have to look beyond patent law to understand the laws that determine how innovators will pay for the development of new technologies, even within the patent law heartland of biopharmaceutical innovation. And I think I'm going to stop there and ask for questions.
We definitely have time for questions, so if you could raise your hand and I'll call on you. I would describe how our new system works, except I don't know, in terms of the microphones. Yes. Can you go ahead and press the button right there? Thank you.
I'll hold it down. Okay. Have any academic studies been done to determine whether there has been a significant effect on patenting activity as a result of Mayo and Myriad?
I don't know the answer to that question. I don't know the answer to that question. Academic studies, does anybody else know about academic studies that have been done to assess this? Not that I'm aware of. There may well be. It might be an easy enough thing to do. I mean, a lot of the patents are available online, obviously, but I don't know. I think the patents haven't gone away. There are a lot of patents out there, particularly a lot of the biomarker patents, but people are afraid to assert them because they'll be held invalid. So you can't necessarily tell on the basis of the universe of issued patents what is going on. But I don't know the answer to that question.
Other questions? Yes. Is it possible for you to press the button down? Thank you.
Has the NIH involved itself in this issue in any way?
Has the NIH involved itself? Well, the CMS has sought to bring NIH into this issue, actually. In the CED part of their decision, they've said that, first of all, they've said they won't cover any test that isn't registered with the National Cancer Institute that maintains a registry of genetic tests. And they've also said that under the CED part, that it has to be basically an NCI network clinical trial or registry in order to get coverage with evidence development. So they are sort of trying to bring NCI in. I haven't looked actually to see if NIH or people from NIH have submitted comments to CMS or anything like that. Certainly there's a lot of, there's some money there, there's some funding there, although not as, they're feeling beleaguered right now by the budget process. That they have some interest in identifying and understanding these genetic mutations, and so you might expect them to play a larger role in at least the CED side.
Thank you for that question. Other questions? Yes, it's possible to press the button down. Thank you.
You mentioned that in light of the fact that both the Prometheus and the Myriad decisions have been broadly read upon, you do believe that the court is confused, and indeed there is human intervention. So what would prompt a changing or narrowing of that broader view?
Well, I mean, don't laugh. In theory, Congress could change the rules by, holding hearings. I know, it's pretty funny. But, I mean, when the court does something that may be ill-considered based on its own prior decisions from many decades ago, and times have changed, one thing to do is ask Congress to change the ground rules. I think that's, even if we had a functioning Congress, I think that would be unlikely in this case because I think they are really low. They have never changed the rules on patentable subject matter. They are just really reluctant to change Section 101 of the Patent Act for reasons that I don't fully understand. But in theory, that's where that could be where it would happen.
Other questions? Yes, there's a microphone coming your way. Thank you.
Hi. You thought at all about why FDA is willing to issue approvals or clearances of these LDTs at whatever level the company making them is trying to ask for?
Well, they aren't necessarily.
That's what's created the.
They were willing. I mean, they are not necessarily willing to. They were willing to. So they had two applications before them. One sought approval as a Class 3 device, which is the most rigorous regulatory approval. So why wouldn't they say that's fine? The other one had to persuade them that that they should reclassify this as Class 2. That this isn't really so risky, and that you can keep this. You can ensure safety and effectiveness for a product of this type with the lesser burdens that are associated with class 2 devices, and they agreed. They decided that they agreed, and they specified very clearly what information they wanted included in the pre-market notification. And if they had thought that, that this was a really risky product, they wouldn't have to have approved that. So I don't think that they were necessarily reflexively approving. Perhaps they were. Perhaps they were thinking, we want these to come to us so that we can see what's going on, and if something is a big problem, then we want to have an opportunity to weigh in and express our disapproval, and maybe even deciding that we're going to exercise the discretion, we're going to change this discretionary move, and we're going to exercise the authorities that we already have to regulate these products more vigorously. So they might want to welcome these applications so that they have a seat at the table in establishing what it takes to establish clinical validity for these products. They might want a larger role for themselves, and they may therefore want to be friendly so that more tests will come their way. And then if they see some fly-by-night operation that really shouldn't be having its test approved, they will have an opportunity to explain why that is so and to issue a decision that provides guidance for other firms. So that makes sense.
Thank you. Yes. Can you press the button down? Thank you.
I might be completely off base here, but it seems to me, all this talk of risk, I mean, these tests have no risk at all, right? So
Well,
this isn't even a legitimate function of the FDA.
the risk is not this. The risk to a diagnostic test would be that it leads to an incorrect diagnosis and therefore to a harmful treatment, right? That would be the risk. So if these tests lacked clinical validity, they might, particularly for cancer care, most cancer care is toxic, and most cancer care is ineffective. So most, cancer patients are receiving care that is hurting them, and if an inaccurate diagnosis might lead to more toxic and ineffective care.
I understand. Thank you.
Okay.
I think we have time for one more question, or two more. Yes, please.
Hopefully I'm pressing the right button. Can you go into a little bit greater detail as to why you think the Mayo decision was off base?
I don't necessarily think it's bad policy. I want to be clear about that. I have to have some. I take very seriously the fact that the academic genetics community thought this was a wonderful decision. Are you asking about Mayo or about Prometheus? About the Myriad case?
No, the Mayo case.
The Mayo case. Oh, the Mayo case, I think was, I just think, what kind of a natural law is that? To say that, here's the things that don't happen in nature. Treatment with thiopurine drugs. Measuring, so patients don't metabolize thiopurine drugs in nature. People don't measure metabolite levels in patients in nature, and they don't, on that basis, adjust the level of treatment. The idea that that's a natural law, what happens, when should you adjust the treatment, the dosage of the drug based on how the patient is responding to it, that's not even a clear scientific question. That's a human judgment call, right? How to trade off the harms of, what the underlying disease that you're trying to treat with the thiopurine drugs against the side effects of those drugs is not something nature has any view about. Nature doesn't call something a disease, much less tell you when what is happening merits treatment and when the burdens of treatment outweigh its costs. Those are all human judgments that are part of the human technology of medicine, and I think it's just whack to say that that's a law of nature. Now, they might still, they might still be right as a policy matter that these patents were getting in the way of diagnostic test development. It may, we've seen a proliferation of new tests following these decisions, and so perhaps they were, they were right as a policy matter. The analysis, I'm really quite certain, is wrong.
Let's take one last question, please. Thank you.
Okay. Company. Oh, okay. If a company goes the 510K route and gets approval and then doesn't ultimately get CMS coverage, is it possible for them to go back and and do the pre-market approval, or is it too late now?
Could Sloan Kettering go back and seek pre-market
Right.
approval? I suppose they could, sure. What they would have, what they would do is they would say, oh, we only got approval for use of this as for tumor profiling, let's get approval I think probably what they're doing, I know actually what they're doing right now is trying to get CMS to change their policy, and that would probably be the better way to go. But yeah, they could go back, and certainly other test developers would go back. So they could seek approval for the same product as a companion diagnostic, which is the strategy used by, Foundation Medicine is basically, it is largely owned by Hoffman LaRoche. It's basically, and they do a lot of work with the pharmaceutical industry, so they're very focused on the companion diagnostics, and they were thinking about it that way, whereas Sloan Kettering is a, cancer research center, and I think they were just thinking about it differently, and they thought that less onerous regulatory pathway would make sense. And it would make sense. That's the real shame of this. Yeah.
Let me say three things. First, we can have more questions, but we're going to do it out in the Zilber Forum. We will have a reception there, so I hope that as many of you as possible will join us. Second, I want to thank all of you for coming out on this snowy afternoon, or now evening, in Milwaukee. We invited someone from Michigan so that she would not be intimidated by the weather. And third, I want to thank Professor Eisenberg for a most engaging Nies lecture. Thank you so much.